So what I'm going to do today is tell you a little bit about the research that we're doing. And as Ben mentioned, hold on, I have to figure out how to forward these. Okay. As Ben mentioned, we were up at University of Alabama main campus, and I have this big lab full of awesome students. And so what you're going to see today is really their work. And it's primarily the work of Shelley and Anthony here. But these are graduate students and undergraduates who have helped along the way in the field and in the lab. And so really, I can't do any of this without them because I'm stuck doing paperwork and writing all day. And so what I wanted to do today is kind of just to introduce you to a couple stories that center around something that should be really important to all of us, which is climate change. And we all, I'm not talking to the general public here, so I think we all are on board with the fact that things are changing and changing pretty dramatically at that. I think we focus heavily on major weather events and glaciers melting and big droughts and fires. Right? And these are big, broad scale things, but the way I think about it also is what's going on at kind of a micro scale within the environments that our animals live in. And this is a challenge for a cryptic species, as you'll see, like ours. And so there's some leeway in how we can actually go about addressing these questions. We don't have data at the micro scale that's needed, but as you'll see, I hope, we have enough data to generate some really cool hypotheses. And so our lab really focuses on the interface between three major elements of biology. So we have the ecological side, knowing what our animals encounter in the natural world, what's their community look like, what kinds of parasites might they be exposed to, who preys upon them, etc. Also the abiotic conditions that they live in, temperature, salinity, pH, and so forth. And I just saw a lab where you guys can control that in a beautiful way. So yeah, if you're hiring, no, just joking. We also think about this in terms of the distant past. So looking at evolutionary trends over time in terms of habitat use or morphological characteristics, or the evolution of big phenotypes is my current love, looking at lots of traits simultaneously and asking questions about how they move through time. But we also have a pretty big organismal focus where we really like to learn about our animal so that we can put it in the context of its ecology and its evolutionary history. And so what I'm going to do today is show you a little snippet of each of these. Hopefully the story flows. This is the first time I've given this particular talk, so you can let me know. But here's our critter. This is the mangrove rivulus fish. This animal is about this big. It's kind of like the size of a minnow or a bait fish. To me, it's one of the most beautiful fish on the planet. But these guys are all hermaphrodites. I don't know if you can see that. So all of these animals are hermaphrodites, and I'm going to get back to that soon. But know that this is a very weird species, a very weird species sexually in particular. And here's their distribution. Get it. So here's their distribution. What you're seeing in green is mangrove forests that basically are ubiquitous throughout southern Florida, Central America, South America, and the Caribbean. And so that's all the green that you see. And from 2010 until now, our charge has been to sample as many populations of mangrove rivulus as humanly possible to get genetic, environmental data. And so right now, we hit 57 populations. We just got this little dude up here, the northernmost population in its range, a year ago. And so what we do is we go out in the field, we do stuff in the field, but we also collect animals from these populations so that we can do manipulative studies back at the laboratory. Move that away. These guys are also strange in a few other respects. They are euryhaline, meaning that they can exist in loads of different salinities, all the way from freshwater to 80 parts per thousand, which is almost little over double the salinity of seawater. And so these animals can exist in loads of different habitats. When it comes to salinity, they can tolerate hypoxic conditions. Dissolved oxygen can actually go down to 0. We've measured it at 0 in the crab burrows that they are swimming around in. Right. And so oxygen tolerance, another thing, and they encounter some pretty nasty chemicals in the mangroves, hydrogen sulfide, that quote unquote fart smell that you smell when you're slogging through the mangroves. That's all the hydrogen sulfide percolating up to the top. And these guys can handle it. Most fish species cannot. Sorry, every time something happens with Zoom, I have to click back. Okay. And this is another really interesting part of their natural history. These animals living in mangrove ecosystems on the coast experience tidal influx, pretty serious tidal influx in some populations where the water goes out and barely ever comes back in. And so these animals oftentimes find themselves stranded on land. And in that case, sometimes they will go and burrow themselves into these rotting logs. This will become important for us a little bit later, but this is how I first discovered them. So I actually went out in the field based on what was published in the literature, ready to collect fish, and I went in the water, as you normally do when you're trying to collect fish, and I couldn't find any. And I sampled for weeks, and I was just getting skunked all over the place. And so back then, I was a little bit more Sicilian and a little bit less chill. And so at that time, I got frustrated, gave up, grabbed this rotting log just to find scorpions or something. That's not what I found. When I opened up this rotting log, out came a bunch of little pieces of popcorn, right? Jumping around. Every one of those is one of our mangrove rivulus fish. And so not only will they occupy terrestrial environments for months at a time, 2 to 3 months they can live out of water in a moist environment, but they also have these great ways to navigate on land. And so this is a high-speed video, super slow-mo. I'm going to fast forward it. And this is an animal on mangrove substrate, just being probed a little bit. And this is how they locomote terrestrially. So they jump, they turn themselves into a projectile, essentially, and use these modified bones in their tail to propel themselves off the substrate. And some of them can jump like 16 to 20 times their body length. So this is not just some random thing. They have been selected to perform in this respect, right? And so this is just a little bit of the natural history of our fish. Let me tell you more about its environment and its habits. And so most animals in a population, and we're talking 95 to 99% of all of the animals are hermaphrodites. They are also self-fertilizing hermaphrodites, the only vertebrate in the world that does this. And so if you're not familiar with this mode of reproduction, it's what plants use lots of times, what nematode worms use. And so the individual possesses both ovarian and testicular tissue, produces sperm and egg, and can fertilize itself. And so most of the animals in our populations can do that. However, and we still do not understand why, males crop up between 1 and in some populations that are extreme, some seasons, like 48% males. So we have this major gradient in male percentages across its geographical range. If you're interested, the northernmost new populations have like no males. And not surprisingly, since there's no actual sex going on, no outcrossing, those populations are genetically depopulated. Whereas in Belize, there are lots of males, they cross with the hermaphrodites and leads to lots of genetic diversity. So it has population genetic implications, the percentage of males that exist. One side of our research program is trying to figure out why they exist, because in my view, you are a self-fertilizing hermaphrodite, you have reproductive assurance, 100%. You know that you can have kids. The moment you turn into a male, all bets are off. You just sacrificed all of your reproductive assurance. And so we're still trying to figure out why and under what conditions the hermaphrodites change sex to male. And so here's just a little piece of the gonad, just to show you that most of the gonad, so if you orient yourself, the head would be up towards the top and the tail would be towards the bottom. And this is the gonad zoomed in, and all of these things are eggs of different developmental stages. And then you can see located medially here, if you zoom in, it's pockets of spermatogenic tissue. And so normally this is how the gonad looks. It looks like a female gonad from the outside, but once you do the histology on it, you'll find that between 1% and 7% of that gonad is committed to making sperm. But it's a pretty tiny amount. And so what are the implications of this self-fertilizing sort of lifestyle? Well, let's imagine that you had a hermaphrodite and a male that mated. Now you pretty much have a completely heterozygous offspring. And so the way that I'm representing this is that you have your diploid two alleles. One of them is dark red, one of them is pink, suggesting that they have different alleles at that genetic locus. Same with the blue, same with the green. But if you self-fertilize over and over again for many generations, every generation, if you do the math, you cut heterozygosity in half on average. And this is just because of the Punnett square. When you make a big A little a to a big A little a, two of its offspring are homozygous. So you cut heterozygosity in half every single generation. If you bring it out multiple generations, what you eventually get are completely homozygous offspring that have different combinations of the parental alleles. And so what you've now effectively done is generate animals that can, for lack of a better word, clone themselves, but it's not true cloning. This is sexual reproduction. They're still undergoing meiosis and recombination. It's just that the sperm and egg come from the exact same animal. And so to visualize that, we have two sample completely homozygous animals up here. And if they make sperm and egg, the sperm and the egg have the exact same complement of genes. And then when they fertilize themselves, boom, they just put themselves back together again. And so this is really useful in the lab, especially because one of the thrusts of our research program is to understand how the environment influences the development of the phenotype. You can't really know that in organisms that outcross because everybody's got a different genotype. But what you can do in our species is take genetically identical animals and put them in different environments. And if they turn out differently, you know it has to be the environment. It cannot be due to genetic variation. And so this system allows us to explicitly partition genetic, environmental, and genetic by environmental effects on the development of the phenotype. And so this is not what I'm going to be talking about today, but I wanted to introduce you to the fact that this is a powerful model organism for that reason. And so what are we going to be talking about today? Well, I have a few stories for you. And so most of this was driven by a remarkably talented PhD student who is a computational biologist who I forced to go into the field with me to make sure that he understood the ecology of our animal. But this dude loves to sit behind a computer and code. And so I don't have that expertise. So if you have questions about precise coding, I can be dangerous with it. But if you really want to get the real answer, you should probably reach out to Anthony and I can give you his contact information. But the first thing that we wanted to know was, where do these guys hang out? We've already been in the field. We know generally where they hang out, but we can't take point sampling data for all habitat characteristics every single time. And when we tried to install $7,000 sondes in the mangroves, they ended up getting wiped out by a hurricane. And so we found one of them in Louisiana, which was great. Not the mangrove, though. And so we really were interested in whether or not what we saw in the field is what pops out when we look at where these animals are and what the habitat characteristics are. So what Anthony did was to address a few specific hypotheses, like salinity and precipitation would predict the suitability of the habitat. This is largely because we know that salinity is an important thing for these individuals. Even though they're euryhaline, that doesn't mean they like every salinity the same way. Right? So we were interested in where on the salinity gradient these dudes were happiest or most abundant, really. We also mapped habitat suitability across multiple years in the future. So we went out, we used these representative concentration pathways, which are just different ways to look at the rate of climate change over time, and I'll show you that in a second. But we varied this simulation model based on the conditions that would be representative of doing really well in terms of climate change, stopping the production of greenhouse gases right now, versus if we continue to do what we're doing without any change. And then we asked the question whether or not habitat suitability might be location specific. So is habitat suitability or its change over time going to happen differently, for example, in the Florida Keys than it does in North Florida? And so the first thing that we did here was we grabbed all sorts of data from tons of different databases online, WorldClim and so forth. And then we got confused because most climate databases either do marine or they do terrestrial. We work in the intertidal. It's at the interface between the two. And so what we did was we took the terrestrial and the marine variables and we merged them and we extended them into coastal habitat. So I'll show you that in a second, but here is the basic full range of our fish. We're zeroing in right up here in the range of our specific species. And within that range, we had a bunch of different sampling localities. And so at these sampling localities, we had years and years and years of abundance data. So we knew where they were, we knew their abundance, and we knew the habitat characteristics of those areas. And so this is just a visual of kind of what we did. If we were focusing here up in Tampa Bay, we don't have climate data for the intertidal. We have it for the terrestrial, we have it for the marine. Marine considers intertidal terrestrial, so they exclude it, and terrestrial considers intertidal marine, so they exclude it, which is really great for our purposes. Right, but what we did was we basically have the climate data from the marine side and the climate data from the terrestrial side, and we merge it together and extend these abiotic data into the intertidal. And so now we have two main pieces of data. We have abiotic data, temperature, salinity, precipitation, and we have presence data for our animal. And then we can basically just use this program called MaxEnt to do a niche modeling kind of scenario, which identifies the habitat characteristics that coincide with where your species might be. Personally, I think this is kind of a circular way of going about things, and I'd be happy to talk to other people who niche model. But if you already know they're there, and then you use the climate data from where they are, well, you would expect a nice correlation, right? But bear with me, right? Because we mapped this across multiple representative concentration pathways from 2.6, which is basically if we keep doing things to mitigate climate change, all the way up to 8.5, which is if we just let climate change go. And so 8.5 is worst case scenario. This is just a sampling of the data. And so we have this for 27 different habitat variables, but I just wanted to give you an idea of what these data will look like after we simulate the niche model. And so we focused here on salinity, in particular, sea surface salinity of the saltiest month. So this is basically just the water that's going to be inundating the intertidal during the saltiest month of the year. And what you can see is that the predicted habitat suitability is very low from 25 to 35 parts per thousand salinity. And then it jumps to be really high, greater than 35 parts per thousand, which is a little bit greater than seawater. The reason why there's a bunch of graphs here is because we ran this model in a number of different ways with different parameters, just to cross-check that the pattern was always the same. And it is, right? So you're always seeing this low habitat suitability at low salinities, high habitat suitability at high salinities. And this seemed to correspond with what we see. Oftentimes when we sample, it's between 25 and 50 parts per thousand. When it comes to, though, asking questions about habitat suitability and how it will change with time, that's where some interesting things popped out. And so if we take a look right here, this is the present day graph. This is the habitat suitability right now. And so up here in the little armpit of Florida, the habitat is pretty bad, according to the model. The blue indicates very high quality habitat for our animals. So Florida Keys is great. Down near Fort Myers is great. We don't particularly see that, but that's what the model says. And then the yellow is sort of intermediate. Now, if we just kept going with trying to mitigate climate change, we would be here. If it's the worst case scenario, we would be here. And so what happens in the future? Well, at the northernmost part, not much. It was unsuitable. It will continue to be unsuitable, according to this model. What was interesting to me, though, was down south. And so in the Florida Keys, right now, it's really suitable habitat. But as you employ greater and greater extreme climate changes, the habitat becomes less and less suitable for this animal. This is a big problem because now what we have is habitat contraction, right? They're not going to migrate up to the north where we expect mangroves to move over the next 50 years, but they're going to leave our rivulus behind. That's what this model is showing. If they go up to the armpit of Florida, they're probably going to die. But now what it's saying is, hey, if we don't mitigate climate change, a habitat that was once amazing is now going to turn into a liability in the Florida Keys. So we're going to contract their range pretty significantly if we don't mess around with climate change a bit more. And so I think this has a couple of implications, right? So the way that we were thinking about it was that mangroves are what they call an umbrella species. It's like a foundation species in a community. And the expectation is that when mangroves move north, everybody else should move north. That the entire community will stay intact as climate change happens and as these mangroves are pushing their way towards Alabama. But what our data suggests is that that is not true. As the mangroves move north, you will not necessarily get all of the community members. We just saw one of these dudes in a tank downstairs. You will not necessarily see all the community members moving with it. And so I think that that's a big deal in the area of conservation because lots of times people focus on these foundation species and they don't focus on all of the other stuff. They just make the assumption that they're going to go with it. Not true. This is the other concerning part. So we use representative concentration pathways for the year 2050, which isn't too far away. The expectation, though, is that we're going to see more and more of a high magnitude change in climate as time persists. And so you can see that these lines are diverging from one another, right? We found the strong effect that we found even here. When the difference between these representative concentration pathways isn't huge yet. So what the heck is going to happen at 2100 or in 150 years from now? Well, we don't know. And so the hope is that we can start mapping this out hundreds of years into the future. There is limitation to that computationally. So one of these models takes like a week to run. one iteration of it. So yay, years of just watching a little status bar. Okay, so take-home message from there is we know what kinds of habitats these animals like, or at least are found in. We know that if the habitat changes, they're likely to experience range contraction. And so now the question becomes, is what we're seeing in the field actually what the animals quote unquote like? So we brought this back into the lab a little bit. These are just cool pictures of the animals that we see in the mangroves, just to hold your attention. Right? And so if you actually go to some of our field sites and you zoom in, you'll be able to catch fish in all of these little crab burrows spread out throughout the mangrove. And what our interest was is how much microhabitat variability do they experience, and do they have a preference for the microhabitat that they hang out in? And so this is that same area, but now you see the crab burrows, and the numbers are salinities associated with that crab burrow. This is a very fine scale. The scale bar is 50 centimeters, right? So we're really only dealing with like, what, 300 centimeters here in squared. But you can see that it ranges from 23.6 parts per thousand, 41.7, and everything in between. Tons of microhabitat variation here. And so our question then becomes, when the animal is faced with this microhabitat variation, what does it do? And of course, there's physiological implications for which salinity you choose to hang out in. You could be in a hypertonic solution, way saltier than your body, and you might shrivel up like a raisin and die. Or you might be in a hypotonic solution and you take on so much water that you explode, right? Or you could be an isotonic solution, which is pretty much the same salinity as your body, and then it's kind of like an equilibrial state. And so organisms have to have ways to deal with the influx of water or salt into their bodies. And this is what drives a lot of physiological variation in marine fishes, especially. And so here was our basic framework. If you are the fish and you are encountered with various microhabitats with different salinities, and you don't like them, well, you have a few options. One is to hang out there and die. One is to get out of Dodge. And one is just to, when you get out of Dodge, find something that you actually can deal with physiologically. So to do this, we came up with a modified, it's called a salinity apparatus. This is a way to maintain a salinity gradient in the tank where the fish can swim through it. And so I'm not going to go through all the details, but the idea is that each one of these little boxes you fill with different salinities, and there's a barrier initially, and then you remove those barriers, and there's only this amount of space for that water to actually mix. And when the fish moves, it moves up and over that little partition, but it doesn't mix the salinities significantly enough for them to change over an 8-day period. So we can maintain this salinity gradient for a pretty long period of time. And now the question is, where do they go? You plop the fish in here, where do they go? Do they hang out at 5 parts per thousand? Do they go over, like our model suggested, to 35 and 45 parts per thousand? Without any other constraints, what do they do? And the data suggest a couple really cool things. So we'll start here. So this is just their preference. And recall that in the control, well, actually, I didn't tell you this, but in the control, it's just 25 parts per thousand across the board. In the experimental, we have that 5 to 45 gradient. The 0 here is 25 parts per thousand. So in the control, they just hang out in the middle somewhere. In the experimental, they are actually hanging out in fresher water than we would expect. That's what the negative value is. The strength of preference was higher in the experimental as well, which is basically just validating our design that we did something with the gradient and they're responding to it. But over on the left is what kind of got me interested. In the control, you see that the animals spend most of their time on the edges. And so that caused me significant intellectual grief because then I was like, well, wait, then are they preferring 5 or 45 parts per thousand at the edges? Or is that just because they like edges? And so we spent so much time on this experiment and we didn't even think about that. So to deal with that, oh, wait, I have one more thing before we deal with that. So it's not just their preference that we measured, it's also where they like to lay eggs. And so here's our control, and you still see that U-shaped pattern. They like to lay at the edges. In the experimental, they like to lay their eggs in fresher water than we would expect, way fresher. 5 ppt is where they laid most of their eggs. Not just that, but you have higher hatching proportions at lower salinities. So now what we have is they prefer lower salinities, they lay their eggs in lower salinities, and the eggs do better in lower salinities. It's a fitness implication. But we find them at high salinities, right? So this is really confusing. So this is partly why we went back and tried to deal with this edge effect that we saw. And we did that by 3D printing circular salinity gradients like this that we can put together in like a little puzzle. And then the fish can do the exact same thing and swim through it, but now we don't have any edge effects. So what happened here? Well, in the control, where it's 25 parts per thousand across the board, we pretty much see that they're spending about the same amount of time in every compartment. However, when you have the gradient, now you are seeing what I would consider a real trend, and it shows the same thing, that there is a very strong preference for fresher water in mangrove rivulus. We also compared the males to the hermaphrodites. This actually went against our prediction. We expected males to not care at all and hermaphrodites to care a lot, but we can get into that later. They both cared a lot. They both like fresh water. And so that brings us to another question, which is, if you are in nature hanging out in way higher salinities than you should be, are there costs to that? And we were focused largely on physiological costs. And so what we did was this massive experiment involving 3,000 animals, where we took, remember, we can isolate genotypes, right? So we have 36 genotypes. Each of those genotypes can give us genetically identical offspring to themselves. So we had 4 replicates per genotype. They're all little clones of themselves. We had 7 salinities ranging from 5 to 65 parts per thousand. And we had 3 life stages: hatchling, juvenile, and adult. And our goal was to plot them in these environments and just see what happens in terms of mortality, growth rates, morphological features, reproduction, and physiology. So the first thing that struck me was that for juveniles and adults, which are these 2 on the right, basically they didn't care which salinity they were in. Adults survived almost 100% of the time, regardless of whether they were in 5 or 65. Dropped a little bit at 65, which is to be expected. Juveniles were a little bit more sensitive, but still 80% or more of these animals are surviving. But hatchlings, this stage of ontogeny seems to be super sensitive to changes in salinity. And so what you see is like a really nice gradient. 5 parts per thousand, nobody died. Then you start getting some death at 15, 25, 35, 45, 55, 65, and it drops precipitously as you increase the salinity. And so here again, we are seeing a situation where they are doing poorly in the environments in which we find them in the field, right? This is how poorly. These are all animals that are exactly the same genotype. They are exactly the same age, but they were placed in different salinities. And so this dude is clearly tiny. This one's a big honkin' hermaphrodite. And so this to me was really striking because it suggests that the animals exposed to these higher salinities are in fact experiencing significant costs. Otherwise, they would dedicate lots of their energy to growth and reproduction, which they are not. And so what we then went to do is use our expertise in histology to look at lots of these different physiological traits related to salinity challenges. And I'm not going to show you the data right now because it's a lot. But what I'll show you are some cool pictures. So what we did was we took the animal's body. This is a decapitated fish here, so its head is supposed to be right here. And we have a liver right here just to orient you. This is the gut. This is the spinal cord. These little diamond-shaped things are the vertebrae, and then everything else around it is muscle. And we can zoom in on, for example, the kidney tissue, which sits right below these little diamonds, and quantify the number of glomeruli, which are the interface between the blood and the urinary bladder, essentially, and asked how big they were, asked how big the capsule that they sit in is. And then we continued to ask some really interesting things like, what's the diameter of this glomerulus? We also looked at the gut. Gut plays an important role in osmoregulation because you have things like these guys, goblet cells, that are filled with these receptors or transporters called aquaporins that move water in and out kind of on demand. And so we counted the number of goblet cells as well. Then we looked at whether there was a trade-off with reproduction. So the idea is that if you are investing heavily in the physiological machinery needed to deal with osmoregulation, you will not be able to dedicate a lot of energy to your gonad. And not to ruin the surprise, but I know I'm not showing you data, but the animals that are in high salinities have like no eggs at all. And if they do, they're teeny, teeny, tiny. Not the big vitellogenic eggs that are stuffed with yolk. Right? So these guys are suffering from a reproductive standpoint at higher salinities as well. But this is my favorite. We just started doing this in the lab. And so what we're doing is we're targeting cells that produce sodium-potassium pumps in the gills. We know these sodium-potassium pumps are critical for moving ions across the gills and making sure that these fish can osmoregulate properly. And so this is the head of the fish. This is its eye and its brain, and then the gills are right here. And what we've done is stained the ionocytes, these cells that contain the transporters or the channels that move ions around. And so we're currently right now at the stage of quantifying all of this histological data. And the idea is to generate a full vision of the phenotype by incorporating the physiological data with growth and life history data, with behavioral data at all of the different salinities to see how the whole multivariate phenotype shifts as the environment shifts, which has evolutionary implications and also implications for example, how these animals might respond in a phenotypically plastic or flexible way to environmental change. And so I'm going to end with just a little bit more of our modeling stuff, because I think it's going to wrap this story up at least a little bit. And so right now, just to remind you, we have an organism that lives in mangroves that experience tons and tons of variation in salinity, lots of microhabitat variability. We find them at higher salinities, but we've demonstrated over and over again that they do not like that. They prefer lower salinities. They lay their eggs there. The eggs do better there. Their gonads are bigger at lower salinities. They are bigger. So everything just points to the fact that where we find them is not ideal. And so we then started to think about this idea of the fundamental versus realized niche, which I think you all are familiar with, but just as a backdrop, the fundamental niche would be like what we did. We look at habitat characteristics, all these abiotic variables like temperature and precipitation, and we see where they could be. That's the fundamental niche. The realized niche is where they are. And this is influenced not just by the abiotic factors, but also by parasites and predators and competitors. And so what we wanted to do was to take the data that we had collected over a long period of time in the mangroves and use the data for all of the species that we collected, which range from like little gobies to mangrove mosquito fish to a bunch of other killifish species and their predators. And so we had to kind of pare this down a little bit because we didn't find a lot of these species at every single site. And so what we did was we grabbed the species that were prevalent across sites so that we could test hypotheses about how the prevalence of these species affects where our mangrove rivulus is. And so zoom in with me a little bit. And so what we basically did was at each site, we went 100 days into the past, and we took all of the climate data for 100 days into the past of when we did the sampling. And so all of this is kind of scaled to the actual events that were happening climatically in that area for the 100 days preceding our analysis. And the reason for the 100 days is because this is about a generation time in our fish. And so we were trying to look at variation within a generation. And what we did was grabbed a bunch of these climatic data again from things like PRISM. We used some HICOM data for the marine variables. We also used some USGS resources for topography. So we calculated what we call vector ruggedness, which basically just how much the substrate is kind of rugose, going up and down. Our fish love habitats where there's lots of little hiding places. And then we did tidal predictions to try to figure out the frequency of inundation into the mangroves at that site for the 100 days prior. And so we have two kinds of things that we looked at. One are detection covariates. And so this is basically just what kind of trap did we use? How likely are we to detect these dudes? And so we used your typical G traps, minnow traps, and cup traps, which are homemade. Funnel and a cafeteria cup that I steal. And so where we deploy these, the G traps or the minnow traps are deep. They capitalize on the deeper water microhabitats, but the cup traps capitalize on upland or shallow habitats. And so let's just take a look at the things that mattered in terms of climate or those traps to our fish. And so let me orient you to this. These are all the different variables that we looked at. The inclusion probability, if it's blue, it just means out of all the simulation models that we ran, this variable kept popping up. If it's red, it means it popped up in a few, but not a lot. So we can have way more confidence that the blue ones are important. The number is the actual sort of correlation coefficient. So it indicates the strength to which that variable predicts the abundance of mangrove rivulus in the field. And so I just want to highlight a couple of things. So here, these variables relate to the distribution of water, and they're very important. They're blue, and they have significant coefficients. So precipitation and tides. Sorry, this is vector ruggedness, right? So this is all about the inundation of water into the mangrove, and that's important for our fish. But the other thing that popped out as very important was this Bungulus confluentus, which is another killifish species that lives in the mangroves, and we often catch them in the same traps as we catch mangrove rivulus. And what this suggests is that anywhere we have abundant Fundulus confluentis, mangrove rivulus populations take a nosedive. And so what we're now thinking is that, oh gosh, let me back up a second. So what we're now thinking is that Fundulus confluentis might be the answer to why these fish are in saltier water than we would expect, given their preferences. Because Fundulus confluentis loves the fresher water too. Just so happens, though, that they beat the living daylights out of our fish. And so if our fish were to migrate into a fresher water area, they would likely get excluded. And so what I think we're seeing here is that competition with Fundulus confluentis is dictating habitat preferences more than their physiological needs, which suggests that this competition is pretty severe. With respect to all of the species, I just want to kind of highlight a couple things. So these are all just important detection covariates. And there's kind of a pattern where the fish that come in with the tides, like Fundulus grandis, a bigger bodied killifish species, they come into the mangroves when tides inundate. Not surprising that change of tide is very important to them, as well as things like G-traps, because they don't typically find themselves in shallow water, they're swimming around in channels. For the smaller bodied species, right, so we have our sailfin mollies, mosquito fish, and Fundulus confluentis. Their abundance is dictated largely by what kind of trap and how many traps we lay. So what that seems to suggest to me is that these dudes are sort of generalists. They get caught in G-traps, they get caught in cup traps, and the number of traps that you lay dictates the number of fish that you get. With rivulus, though, there's only one thing that mattered, which was the number of cup traps that we laid. And this might sound inconsequential, but from my perspective, it really asks the question, do we have microgeographic separation of these species within the mangrove? And they're sort of niche partitioning in a way to reduce competition. So that Fundulus grandis, for example, hangs out in deep, rivulus hangs up in upland, and others migrate between the two. And so this is our general conceptual framework for that, where we think that the most overlap with our fish up here is Fundulus confluentis, driving that competitive exclusion from freshwater habitats. And so I'm going to end with this, and this is just exciting to me. We hang out in these mangroves all the time, and now we're quite interested in how the fish move from one population to another. It doesn't seem like it would be a really great place to move, but these tides do come rushing in and rushing out. And so there's lots of opportunity for them to move, but they're not going to move through an environment like this. If they try, they're going to get eaten by the nearest barracuda or clownfish or whatever, right? And so what we think is going on is passive dispersal. So these dudes tend to lay their eggs on substrate or vegetation or logs. And the idea is that they might lay them on something that floats, and then that will float away to a different position, different geographical locale. But we also know that these adults hang out in logs, like I was showing to you before. And the idea here is that maybe when you're stuffed into a log that's rotting, that floats really well, you just take this like a, what is it? What is the cruise line? Yeah, it's like a cruise line, right? Let's go from Belize to South Florida. Hell yeah, jump in, right? And so as they move off, what we're really interested in is how the ocean currents predict patterns of gene flow among populations, and not just in one direction, in both directions. And so we just actually published the results from this in Frontiers in Genetics. And we basically demonstrated that ocean currents are essential for predicting patterns of gene flow in this species, suggesting that one of those passive dispersal mechanisms is probably the driving force here. And now we're trying to get at more explicit questions related to what that gene flow will do to the adaptive potential of populations. So with that, as we watch the currents circle around the world, I would be happy to answer any questions. And thanks again. I appreciate your attention.